Hi, Michelle Glass here. Welcome back to another episode in our Chapter 8 lecture series. We are moving into part 2. We're looking with this section about our second type of cell division called meiosis, which is our sexual division or sexual reproduction. We have a little bit of terminology to tackle before we really get there. So let's take a few minutes here to talk about this term gene, which we've introduced a little bit before and we'll be talking about again. Genes are our units of inheritance, and you kind of know that already. They're composed of our DNA molecule, and their job is really to code for proteins. So we'll be talking later on in the semester exactly how the cell is going to use the DNA molecule and those instructions in order to build new proteins. When we talk about the term genome, we are talking about all of the genes that are located in a cell. And again, that's talking about all of that DNA molecule. Our DNA, remember, is described as a double helix, where you have those nitrogenous bases kind of at the center, pairing up together based on the rules of complementary base pairing. So we've seen before, adenine pairs with thymine, and cytosine pairs with guanine. So of course, adenine is represented by the A, and thymine is represented by the T, cytosine is C, and guanine is G. We've also described that our DNA molecule in the cell is going to be in one of two forms. So it's either going to be in the chromatin super relaxed form. In the chromatin stage, that's when our DNA is going to be doing the job of gene expression, meaning getting those instructions out to the ribosome to make proteins, and also DNA replication, which we've talked about now as occurring during interphase, that time in the cell cycle when the cell is busy doing its job or preparing for division. When your cell is in the process of division, whether it's mitosis or meiosis, we will see it in the packaged up condensed form, which is described as chromosomes. So that's where that DNA gets tightly wrapped around proteins in order to make sure pieces are not lost or broken in the process of cell division. We've talked about homologous chromosomes before. So in a diploid cell, all of those chromosomes are paired up. They are paired up based on the same kind of length, the same kind of staining pattern, and the same centromere location. Remember, we've talked about homologous chromosomes as being like pairs of shoes, meaning that they clearly match up, but they are different, just like a left shoe is different from a right shoe. Your homologous pairs are different because they're coming from different gene pools, one from mom and the others from dad. When we say that they have the same type of genetic information but not identical, maybe they're carrying information for eye color, which is not inherited by a single gene. So this is super simplified example, but maybe, the dad is passing on the brown eye trait and the mom is passing on the blue eye trait, but this trait is located at the same kind of location on both. Or we've talked about the hitchhiker's thumb. So I have a straight thumb. The hitchhiker's thumb is more kind of bent back when relaxed there. So that is coded for by a single gene. So that is a good example to use here. Remember, our somatic body cells are diploid, meaning that complete set. And we can abbreviate diploid as 2N because that number does vary species to species. For humans, the diploid number is 46, or 2 times 23 because we have 23 pair. Remember, before cell division occurs, whether we're talking about mitosis or meiosis, you're going to have to replicate that DNA during interphase. So this is during that S phase of interphase. So that's where you're going to see the formation of the sister chromatids. Sister chromatids are identical to each other, and they are attached together at centromeres. So remember, we still count our chromosomes based on the number of centromeres. So on each side of the arrow, we have 2 chromosomes, but on the left side, we have 2 chromatids, and on the right side, we have 4 chromatids. So we have duplicated our amount of DNA so that we can now get ready to go through cell division. When you look at a human karyotype, or karyotype for any organism really, what you're doing is taking a cell that's in the midst of cell division. So you've already replicated your chromosomes. We're seeing our homologs paired up based on their length and their centromere location and their staining pattern. So here we're seeing it's a human karyotype, so we have 23 pair of chromosomes. And even though it's kind of hard to see, they've already replicated. Their sister chromatids are available there. So you take a cell that's in the midst of cell division, maybe prophase or metaphase, and you're able to, in the past, you would take an actual photograph of the cell and you would cut out your chromosomes and you'd line them up and paste them. And now, of course, that's all computer animated. When you look at this karyotype, you can see in humans we have 22 pair of what are called autosomes. So these are going to be chromosomes that are coding for all sorts of information across the body. And then we have one pair of chromosomes that's described as our sex chromosomes, and these are going to indicate the sex of the offspring. So individuals that inherit two X chromosomes are female. Individuals who inherit an X and a Y, which is what's pictured here, is male. Notice that that Y chromosome is significantly smaller and has much less DNA and fewer genes than the X chromosome. So this is actually not a homologous pair at all. We will not see crossover happening in that pair. Okay, that's it for now.